now that there is, as Dr. Mitsui and Dr. Friedman yesterday talked about the promise of AI, which there is, and machine learning, and the fact that we know that the pathologists had trouble in that one landmark article about ballooning degeneration. How do we reconcile that with the patients and the stakeholders who come to me and say, you know, Frank, nobody wants to have a liver biopsy? I just want you to comment on that, because I was left yesterday, I heard one of the pathologists talk about the beauty of this tool, of course, for diagnosing tumors, which I would agree with. But I wanted to hear from some of you who are interested in alternatives to histology, what your take is on AI and machine learning, because we consider that also a biomarker, a surrogate. And I will start with Professor Sanyal, and then I want to ask Dr. Morin, and then finally Dr. Mitsui to come back with a comment. Thank you.
So Frank, thank you for the question. I view this as part of our armamentarium and not as sort of a replacement of, you know, a tool and be a standalone thing by itself. So in early phase trials where we have short duration, small numbers of patients, it takes a big effect size to move the needle on conventional fibrosis stage, which captures both the distribution and the amount of fibrosis. Circulating biomarkers, you know, they give us a overall sense of the total fibrosis burden or fibrogenesis, but not necessarily changes in distribution, etc. So in the short term, the AI, where the current data sets, gives us a way to identify changes that may not otherwise be captured by the classical NASH CRN or even the SAF scoring systems. Furthermore, it allows us to capture changes in multiple dimensions. So what I mean by that is that you can see changes in parameters linked to fibrosis in periportal areas, centrizonal areas, perivenular areas, in the septa, all separately. And there's a whole emerging field about reclassifying how fibrosis evolves and resolves in NASH, learning from hepatitis B and other states where there are characteristics of progressive septa versus regressive septa. And certainly with TROPIFEX trial, we were able to see changes using some of these parameters that were not obvious with conventional histologic staging. The key issue then becomes, do those changes really reflect the likelihood that in the long term you will have less progression to cirrhosis? I think we will need more data for that. So I'll stop there and let others weigh in.
Thank you. And Professor Naredny.
I'm just going to build on the same what Dr. Sanyal said, but I'll go with patient's example, and I want to assure them that we're all sides are invested in their best interest. But I will go back to that. I want to answer the machine learning with the patients that I presented earlier that are excluded from clinical trial. I think it's clear today that the same people who implemented NASH resolution and ballooning as a surrogate biomarker a decade ago, now after doing research, and that's what we do in medicine, we do have a problem with the ballooning. And that's a great example for machine learning. That patient was excluded, although the local reading said he's otherwise included. And he's an F3 patient. What do I do with such a patient on the long term? We know weight loss and exercise doesn't work all the time. So the people are trying to push the envelope not to lose such a patient, and especially in the controversial area. And I consider that ballooning cell controversial now at least, and we need to resolve it. Is the question, we go back to 2 points NASH improvement? It could be. That's something we need to discuss. Along the same line of what Dr. Sanyal said, I will remind you, multiple people, Dr. Rocky, Dr. Friedman, in this workshop, they said, I don't need necessarily the patient to improve. We all know the concept now. Well, we're sure we need them improved, but we know the concept now. If you can hold the cirrhosis for a very long time, or do even tiny changes, we can get better. And F3 to F2 is not like F4, F3. So that's where we're talking about machine learning. If I can go from F4 to 3.75, I just made that up, although there's literature on that, that's a win. And the current system does not allow us to do that.
Thank you. Professor Ratsu, any final comments? I want to go with one more question where we only have about 9 minutes left. So I want to go one final question that this leads into this. Do you have any comments, Professor Ratsu? Follow up.
Yes. So your question, Frank, was...
Well, I think that we talked about tensions between, not doing biopsy for all the reasons that we've discussed at this meeting, and yet the promise of AI. You mentioned it yourself. I mean, how do you reconcile that with patients and other stakeholders who are not wild, obviously, about doing liver biopsy? And I have a follow-up question I want to get to that I'm going to go around and ask you all. But if you have any comments about this, if you don't, that's fine. I'll move on to my next question.
I have a short comment on that. It is clear that digital pathology holds great promise, and Arun explained very well that it provides a better understanding of the kinetics of fibrosis changes, both ways, regression and progression. After all, these fibrosis stages, whether METAVIR or NASH CRN, they're very skewed in favor of architectural distortion rather than the amount of collagen. And what you want to understand in the beginning is not just that sort of surrogate of fibrosis, of extracellular matrix deposition, but rather a finer understanding, more subtle understanding of the ins and outs of how fibrosis progresses, regresses, the quantity of fibrosis, the type of fibers, and so on. And of course, that is useful in the beginning of a trial when you investigate the potential of a product. It's not so much useful for large phase 3 trials where you have to show clinical benefit. So, but the great potential for digital pathology. But in order to do that, you need to do a liver biopsy.
Oh,
You're
yeah.
saying a lot of people are not happy with liver biopsy. Well, you have seen at the division now phase 3 trials on thousands of patients that had 1, 2, or 3 liver biopsies performed. It is possible to get those liver biopsies done. It is possible to convince the patients that it is important because people desperately need drugs for this condition.
Okay.
The way I'm ending just by saying that the way I outlined that in the beginning, saying that phase 3 trials should probably be performed with a mix of biomarkers rather than relying on histology, makes it that we will save a lot of these biopsies in the future if we adopt a paradigm where biopsy would only be restricted to early phase trials for the reasons we've seen, but not for phase 3 trials. So it's already a big step forward by saving all these biopsies.
All right. So my final question, thank you, Dr. Red. So one more question, just briefly. So we've heard a lot at this meeting about NIT. Certainly, I've learned quite a bit. I guess one of my questions that you can all put a regulator hat on, and Dr. Laura Lee Johnson and I will sit back and listen. Our draft guidance compared to the European Union, I don't know what the Brits do now because I know that they're separate, but we have these 2 endpoints that we've established, as you all know. And I guess one of my conclusions would be from all of you, you can play the regulator. If we dumped the no reduction in the NIS score and just focused on fibrosis, since the vast majority of what has been presented to me, at least, maybe I'm wrong, in the past 2 days has been squarely focused on fibrosis. And as you all know, someone who studied fibrosis in the laboratory for 20 years, it's near and dear to my heart. But now I'm a regulator. So you be the regulator and you tell me, Frank, it's okay if you and the EMA dump the inflammation. And we'll worry about that because we're just going to look at fibrosis because to me, it seems that the NITs that have the most robust data, Quentin and Arun, are those that really squarely focus on the fibrotic response of the disease. So I want you to answer, and I want to let Dr. Ansley talk first because he hasn't spoken about, and then Dr. Sanyal because we'll have to wrap it up. But I really want to thank you. These questions have been on my mind. They're sort of out in left field, but I wanted to bring them up. Thank you.
No, I think this is an important point, and it's undoubtedly true that in terms of NITs, the strongest NITs we have are for fibrosis. However, the key thing to remember with NITs is that you have collinearity between the inflammatory processes, in other words, the steatohepatitic processes, and the fibrosis processes. And that is the case even, if you think about histology, it's very seldom you see an active progressive fibrosis without the presence of steatohepatitis. The same carries through into NITs and so on. I think it is right that we should be focusing on fibrosis because it's actually the thing that we know is the closest to hard endpoints. But, and this is the key thing, Frank, here, this is the area where MOAs begin to come into the discussion because there will be some anti-metabolic drugs that work a long way upstream. And so they would take an unmanageable amount of time to read through into a fibrotic change. So I think here you've got to tease this one apart. I completely agree. There isn't an easy answer.
Yeah.
But
short answers.
But what are the solutions? Well, solutions are, if we're going to stick with histology, we need to move away from ballooning yes, no, and consider it a quantitative change. AI approaches can help us do that. So that's the first thing. The second thing is we need to cross-validate. And with that Venn diagram I showed you, 2 of those as a minimum should be NITs. You can consider whether the third one is an AI-assisted histology or not. But as a minimum, 2 of those 3 circles should be NITs, would be my suggestion. And that gives, with my regulator chapeau on, that gives you the degree of security.
Thank you, Professor. Dr. Sanyal, your thought.
Very quickly, in the interest of time, I agree with what Quentin said and would like to reiterate the issue of mechanism of action of the drug. So if a drug works primarily by reducing activity, you should include a measure of improvement of activity. And it may be that you have to use a combination of biomarkers to holistically assess that. And to the extent that we can use biomarkers that are multimodality, like one, for example, measuring stiffness, and another one measuring a biochemical measure of injury, etc. Since none of them are perfect, it'll give you a better idea it'll make you more reasonably certain that this will translate into something beneficial.
Thank you, Dr. Sanyal. And Dr. Naredin, and we'll get to everyone else. Yeah.
Sure, I'll do it quick. I think we don't want to remove disease activity per se. Our issue, I think, is with the NASH resolution and the data that has shown it has caused problems. To quick answer, if we would stick to histology, I would keep the fibrosis improvement, as you said, and personally, I would rediscuss 2-point NASH improvement and address the weaknesses in ballooning. That can be done through new methods that innovative and measure tiny changes such as machine learning. Nevertheless, my last 30 seconds is, I think I do respect what you guys do. I thank you for doing this. I understand the responsibility, but we also would love to sit down and talk about what is reasonably likely to predict outcomes. We do personally think that NITs are there. We do also think the NITs can measure disease activity if you combine them. And I think this is the core of the discussion that we need to continue.
And thank you. And you have about 30 seconds each, Professor Atsu.
Yes, Frank, I'm very happy you raised this point. As currently defined, the composite of NASH resolution without worsening of fibrosis makes sense, because we know that as people progress towards cirrhosis, the inflammatory activity might disappear and therefore might have less steatohepatitis activity. That makes sense. What, in my opinion, doesn't make sense, and I think it addresses your precise question, is the composite of fibrosis reduction without worsening of steatohepatitis. That doesn't make sense. It might theoretically fit with some drugs, but we never see that in the natural history of the disease. So I think clearly that if you keep the histological, based on conventional pathology, definition of this endpoint, you should get rid of that part of no worsening of steatohepatitis. So just fibrosis reduction, one stage, and that will help us make comparisons that are more valid between studies, because some people define NASH resolution in different ways, as you very well know. So that makes no biological sense to me, and I think we should get rid of that part of the definition.
Thank you. And finally, the last word from Paris, Professor Castera.
So in a nutshell, get away from ballooning, focus on improvement on fibrosis, use at least 2 unrelated NITs, and LSM should be at least 1 that should be used because we have the highest level of evidence.
And with that, I want to thank you all. Dr. Miller, you can conclude the meeting. I do want to thank you. These were very fun, provocative questions. So we appreciate your input.
Thank you very much to everyone for this wonderful panel discussion. Honored to co-chair that, co-moderate that with you, Frank. And we have lots to talk about as we move forward. And